Please help me welcome John Monyror to the stage.
Day-to-day life depends on systems that we don't really think about until they stop working. When I was a kid, my first dream job wasn't firefighter or scientist. I wanted to drive a garbage truck. I loved watching it pull up to our apartment building like clockwork, this big machine lifting heavy bins, keeping the whole complex clean and functional. Now one day, the driver of the garbage truck waved to me, and I felt like I'd been acknowledged by the most important person in the city. I'm serious, this guy was a superhero to me. I'm sure some kids hope for a wave from Connor McDavid, but I was over the moon with a wave from my garbage truck driver. Unfortunately, my career took a different turn, so let me direct you to this graph over here, where on the x-axis is my life over time, and on the y-axis is nerdiness. I think I used to be a pretty cool kid. I mean, look at that jacket, look at those jeans. It's undeniable. But then I started to wear some button-ups, and then eventually I got a pair of glasses and it was over. I became a nerd. I found my love for neuroscience, and eventually I found my way into the Scipione lab to do my PhD, and we study neurodegenerative diseases. So these are diseases like Alzheimer's, Parkinson's, and Huntington's disease, that at face value, we think of as very distinct from each other, but in reality, they all share something in common, and that's that all of these diseases are driven by the misfolding of a protein. So in each of these cases, a protein misfolds, becomes sticky, clumps, and accumulates. Our lab in particularly studies Huntington's disease as a model for all of these diseases. And Huntington's disease is caused by a mutation in the Huntington gene that results in the misfolding and aggregation of mutant Huntington. And in small amounts, cells can sort of manage this, but neurons are long-lived cells, so over time, they eventually succumb to the burden and they die. Now, we're particularly interested in studying the most basic levels of these diseases, so we can't really do our experiments in humans, so we have to turn to an animal model. And it turns out that mice have almost the same Huntington gene as humans, so we can go in and replace their gene with the exact mutation that we know causes the disease in humans, and we can observe the effect. So here on the left, I'm showing you a healthy wild-type mouse that can cross this narrow beam pretty easily. And on the right is a Huntington's mouse. I think the difference is pretty obvious. Since we know the exact cause of Huntington's disease, there have been two obvious targets that most of the field has focused on. One is to target the gene, the other is to target the protein. Now, gene therapy on paper sounds great, but unfortunately, years of work that have led to great strides just haven't gotten us there yet for Huntington's disease. Similarly, others have tried to directly degrade the misfolded protein or promote the degradation of the misfolded protein, and this has also not led to any broadly effective therapies. But we think there's another way to think about this problem. So here I'm showing you a neuron, and if we zoom in on this really important structure called the plasma membrane that separates the cell from the outside world, we see that's made up of all sorts of components, lipids, proteins, and sugars. And we think that this structure is really important for the handling of these misfolded proteins. So let's zoom back out just a little bit. So when cells start to fill up with these misfolded proteins, they can get rid of them in a pretty interesting way. First, that plasma membrane folds in on itself and creates these organelles called endosomes. As those endosomes mature, their membranes also fold in on themselves and they form these small internal vesicles. As those vesicles form, this misfolded protein can get packaged into those vesicles. And then the cell does something kind of surprising, which is it fuses these endosomes back to the plasma membrane and spits out all those vesicles into the extracellular space. And we call these extracellular vesicles, or EVs. Now, one thing that we and others have observed in Huntington's disease is that the amount of these vesicles that get secreted is reduced, and we think this might contribute to the accumulation of these misfolded proteins inside of neurons. So if we zoom back in to that plasma membrane, there are these really interesting glycolipids, or these fats with sugars attached to them, called gangliosides, that are present on the membrane. And they're really easy to overlook because they're not the usual suspects, genes or proteins, but we know that they play important roles in membrane organization and cell signaling. And one thing that our lab found that's an underappreciated phenomenon in Huntington's disease is that the levels of these gangliosides decreases. So we wanted to know what the consequences of this was in using cells in a dish. And what we found is that if we deplete gangliosides from cells independently of Huntington's disease, we see a reduction of the secretion of these vesicles. Similarly, if we look at Huntington cells, we see fewer of these gangliosides and fewer EVs secreted. But if we were to replenish gangliosides in cells, we see more of these EVs secreted, and they're filled with misfolded protein. Now, this is where the garbage truck gazing younger version of myself gets really excited. So one can imagine the neurons as a house that's producing tons of trash or misfolded proteins. And then gangliosides come in as my hero, the garbage man, and packages up these misfolded proteins into the cell's garbage bags or extracellular vesicles. And when this system works better, the cells look healthier, there's less mutant huntingtin inside of the neurons, and the cells look less stressed. But of course, getting the misfolded protein out of the neuron is not the same as eliminating it from the brain. So where does it go? And this is where we think that gangliosides might play more than one role. We think that the gangliosides that get enriched on the surface of those vesicles can target them to these special cells in the brain called microglia, which are the brain's immune cells and can act as scavengers, coming in and eating up all of these vesicles and degrading that protein in a much more efficient way than the neurons can. So a lot of this mechanism is still being worked out, and a lot of the work I've told you so far has been in cells, but it opened our eyes to a pathway that we hadn't been thinking about enough. So then we wanted to see what would happen if we did this in our mouse model. So here, again, I'm showing you a Huntington's mouse, a control mouse, that struggles to cross the beam, and eventually he falls off. Poor guy. And on the right, I'm showing you a mouse where we can implant a reservoir full of these gangliosides and infuse them into the mouse brain continuously. And as you can see, it pretty much completely corrects the dysfunction. And if we look in the brains of these mice, they also have less mutant huntingtin. And this is what's really exciting to me. Something that's so easy to overlook, a membrane glycolipid, can correct the protein burden of misfolded huntingtin, but also correct the function in the whole animal. So this brings me back to that garbage truck driver. I don't think young me was too naive to think that the garbage truck driver was the most important person in the city. When pickup stops, trash piles up and the whole city would feel it. So even in diseases where the target is obvious, we can't afford to focus only on the culprit. We have to understand the systems that quietly determine whether the cells can cope. Thank you.